So I'm just going to kind of go read through this article. I think this is interesting because a lot of times we think that lung cancer is caused by smoking too much. And back in I think it was the 1930s, the early 1900s, the link between smoking and cancer was found when they basically took like this tar, the tar that is present and aerosolized when you're smoking, and they like blew it into these like cow lungs. And over time, the cow lungs or was it? No, I think it was a mouse experiment. I'm forgetting exactly the study. They had an animal model and they were like putting the tar that's from the cigarette smoke onto the animal or into the animal's lungs. And then they saw that there was formation of cancer. And that was kind of an early indication that smoking can result in lung cancer. And then, of course, there's been a lot more data showing that link. But there are a subset of people who specifically get, let me see, can you guys see this text? They get non-small cell lung cancer, or NSCLC, and they have never smoked in their entire life. And so the big question is like, well, why are these people getting lung cancer still? Like, how is it possible? So one of the sort of hypotheses out there is that maybe it's air pollution. Now, interestingly, like one question I have is, well, do we see higher rates of lung cancer in areas with more air pollution? So I found like this sort of review, and I don't have the link up right now, but they were basically looking at areas that have a lot of air pollution, and they saw that those people who live there have higher rates of lung cancer, and they also have higher rates of like other like cardiac issues and like respiratory issues. But they did see a higher rate of lung cancer in the people who lived in the areas with higher air pollution. So that was kind of a clue that maybe air pollution is doing something somehow to initiate lung cancer. And so this study was trying to figure out like, well, how could that even work? What's the mechanism at the cellular level? So I'm going to read through this bit. Well, so this paper, the study is performed in the UK, and most of the people that they're looking at are either people who live in the UK or people who live in Taiwan and other, I think like there was another country that I'm forgetting. But anyways, so in their study, the data showed that exposure to airborne particulate matter that's less than 2.5 microns in diameter. This is kind of important because there is a distinction where larger particulate matter can have maybe a different effect than the smaller particulate matter. I'm kind of forgetting what the main distinction is. I'm wondering if it's just having to do with like whether or not it can go into the bloodstream. Obviously, the smaller a particle is, the more likely it is it can go into the bloodstream or get like picked up by cells. Their data showed that exposure to the airborne particulates that were very small increases the risk of this lung cancer. Specifically, this lung cancer has these mutations that are called EGFR mutations. So EGFR, it's a it's a receptor that regulates growth of your cells. And when a cell gets a mutation in its EGFR or like EGF receptor, some of the mutations, there's several different classes of mutations, but if you get the EGFR version 3 mutation, this makes the receptor signal all the time. So it's constantly telling the cell, grow, 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 and like survive and proliferate. And there's other downstream effects. And all of these are sort of like pro-cancer functions of the cell. So the cell will keep replicating, and you'll get tumors that have this characteristic EGFR V3 mutation. And so it's kind of like a mutation that gets selected for because it makes the cell keep growing. And therefore you see a lot of cancers that have this specific mutation. Yeah, it's the most common mutation-driven type of lung cancer. Air pollution drives the release of inflammatory mediator interleukin-1 beta. So this is just one of those little cytokines that we've mentioned before. And it's produced by, in this case, it's produced by epithelial cells. So these are like skin cells, but you also have them inside your body as well. And also macrophages, which we've mentioned. So they make a lot of this thing that we're going to call IL-1 beta. And this study found that the IL-1 beta acts on the lung cells that have the EGFR mutations, and it promotes lung cancer development. And in their paper, they go over like exactly how the IL-1 beta kind of, I think what they found in the paper is that the IL-1 beta made these cells with the EGFR mutations more likely to like regenerate and grow, and of course like promote cancer in that way. Sci-Hub, yes. We'll just say it. Sci-Hub. I don't know if I can get in trouble for talking about Sci-Hub, but it is a great resource. Y'all should use it. It's kind of a little bit of a backdoor to get into because they keep having to remove the website. But once you find a way in, it's a wonderful resource. I worked at a place that specifically hired undergrad co-ops, just so they can keep journal access by way of their undergrad library. Wait, oh, like these were undergrad students? Wait, okay. That's interesting. I've never heard of that. I'm not sure if people in industry pay for
now
like
we're ready to head for the highway. Hey, roll and go. Our boots and our clothes, boys, are all in the pond.
Are you trying to cover up what I'm saying about Sci-Hub Nippon? It's from the Crick Institute, which is down the road from my university. No way! Oh my gosh, you're from the UK? That's so cool. Wow, what a small world. Okay, well anyways, moving along. So let's see. Okay, they talk about the particles. These particles, again, are typically found in vehicle exhaust and smoke from fossil fuels. Exposure to these pollutants can trigger lung cancer in cells with EGFR and KRAS gene mutations. KRAS is another really extremely common mutation that again promotes cell growth and regeneration. So these are typically seen the most in cancers because of their like direct function in making the cell grow more and more. And there's other things other than growth that these two genes are responsible for. The EGFR mutations are seen in lung cancers of about 50% of the never smokers, and this new research may pave the way to new prevention approaches. Okay. So the same particles in the air that derive from the combustion of fossil fuels are directly impacting human health via an important and previously overlooked cancer-causing mechanism on lung cells. The risk of lung cancer from air pollution is at least 10-fold lower than from smoking cigarettes. That's good, but we have no control over what we breathe. Globally, more people are exposed to unsafe levels of air pollution than to toxic chemicals in cigarette smoke, with air pollution responsible for over 7 million deaths globally. I don't know if that's annually. I think it might be, but I'm not sure. They don't say. That's something I can look up, but these new data link the importance of addressing climate health to improving human health. The findings are derived from basic and human research on mutations in EGFR and lung epithelial biology. So their study included almost 474,000 people from England, South Korea, and Taiwan to look at the association between exposure to air pollution in these regions and the development of lung cancer. I'm going to just keep saying lung cancer because I always get tripped up over NSCLC. It's hard for me, okay? Increasing levels of particulate matter in that were less than 2.5 microns in diameter were associated with an increased risk of EGFR mutated lung cancer and mesothelioma, among other diseases. Country logic, that's a great point. Who's going to push the outdoor air purification business? There's no way to profit from it, so you know it's going to be pretty hard. I feel like this is just a study that's telling us some depressing news. Like, this is just one more way in which you might be potentially getting effed by, you know, living in a very high pollutant city or whatever. Isn't BRAF the most mutated form of RAF? Oh, I mentioned KRAS. Hang on. Wait, KRAS? Because, well, so KRAS is separate from BRAF. At least that's what I thought. In this lung cancer that they were looking at, they did genetic sequencing of the lung. I guess what did they take lung biopsies from these people? I'm trying to remember. They somehow got lung biopsies from all of these people who no, they had a smaller cohort of people who actually ended up getting the lung cancer, and they took biopsies from those people's lungs, and they saw that EGFR, I don't know if it was V3, I have to check the paper, and KRAS were the most present mutations, or those were the mutations that were making up most of the cells or something. I don't know what I'm trying to say here. They were the most represented mutation. Yeah, the researchers performed ultra deep profiling of 247 normal lung tissue samples and analyzed normal lung tissue from humans following exposures to particulate matter. EGFR and KRAS driver mutations were found in 15 and 53% of the normal lung tissue samples, respectively. This sentence kind of confused me when I was reading it, because I was like, okay, so they had a group of normal lung tissue samples, and then they also had normal lung tissue from humans following exposure to particulate matter. How do you control that? I'm sure they have that detail in the paper, which again, if we have time, I'll pull up the PDF and try to find it. 6, hello, welcome back. What's going on? Oh, yes. So we are talking about the link between air pollutants and small particulates and how they can be linked to causing lung cancer in people who do not smoke. So particulate matter promotes a macrophage response. So this is what they found in the paper. The particulate matter promotes a macrophage response where they make this IL-1 beta and a progenitor-like state in lung epithelium, harboring this mutant EGFR. Consistent with these findings, the exposure to particulate matter increased tumor burden in EGFR or KRAS-driven lung cancer models in a dose-dependent manner. So what was cool is they first did, the first half of the paper is all in humans, and then we have like these mouse models where the mice have mutations in EGFR or KRAS, and they'll develop cancer eventually. But if they expose those mice to particulate matter, then those mice got cancers faster than the ones that were not given the particulate matter. And the reason they do it that way instead of just using like normal mice is because it would take probably forever or longer than the mouse's lifespan for the cancer to actually develop. And also because they knew specifically from their human data that the presence of this mutation had to be already there in the cell for the risk of cancer to actually, or for the potential for the cancer. What am I trying to say? They knew that these mutations had to be present already in the cells that eventually became cancerous. I think that's what I'm trying to say. Yeah. Okay. Let's see. Okay, blah, blah, blah. Okay, their next question is to discover why some lung cells with mutations become cancerous when exposed to air pollutants and others don't, which is, I think, one of the unopened questions that they have from that paper. Yeah. Either way, I think it's really cool because I think this paper was one of the first papers to show that it's actually an immune response in the lungs that is triggering cancer. Usually we think of the immune response as being helpful, although if you have chronic inflammation, we know that this can cause like a buildup of reactive oxygen species, and this might trigger like mutations and stuff in cells, which could eventually lead to cancer if it happens long enough. But this paper was really cool in that they were able to show this link, specifically showing that the particulate matter caused the inflammation that then caused the cancer in their model. Yeah, I'm like, are my words making sense right now? Okay, let's see what's going on. I wonder if this particulate matter affects my adult acne. Oh dear, adult acne is not great. I don't know. That would maybe particulate matter is to blame for everything. Death is an unavoidable side effect of life. That's true too. At some point you're just like, how far can we prevent the onset of cancer? Here's a crazy one for you. You worked at Hanford as a security guard for a while, was exposed to constant radiation for 3 years, and tested early to ensure that I was above the legal maximum in the year. Wait, to ensure that you were above the legal maximum? Do you mean like the legal maximum amount of radiation that you should be exposed to? That's pretty bad. I hope you're okay, 6. Okay. I am going to quickly try to connect my iPad so we can like look at the PDF together, and then I swear I will be done with my science topics for the day because that's what I had planned. But I think the paper is really cool, and when I was reading it like a few days ago, I was kind of just excited by it, and I really wanted to share it with you guys. So give me a second. Take a moment to grab yourself some water. If you've forgotten to drink water, that's important. I'm going to quickly refill my water and come back and connect the PDF, and we'll take a quick look. And yeah. Thanks for listening to my TED Talk. I don't know what you say after that. Weird. Okay, the broadcast has started. I think this is working. Let's see. Yeah. Okay, that is not the paper. Give me a second. So I find it. Swanton. No, wrong Swanton. Swanton 2023. Oh yeah, so this paper actually came out, I believe it was in April of this past year. 2023. It's no longer 2023. In my mind it is. It was published April 2023 by Charles Swanton. There's a huge list of authors on this paper as well. All right, I'm going to try to pop open this window, and now it should be able to recognize it and share it. Let us see here. Thank you, Nick. I'm still not an expert. We have a postdoc in our lab, and she literally can read papers so quickly. Like, these are chunky papers with a lot of sequencing data, and she can read them in like less than an hour and come up with like a journal club presentation from that. And it always blows me away because I take it like, if it's a chunky paper, it takes me a while. This paper that I'm about to read to you, I feel like I was able to understand it during my 30-minute commute and get through the majority of the paper, which is a good sign because before it used to take me much longer. Now, because this paper is not open access, I'm going to try to pop a PDF into our lovely little Discord later so you guys can still see it because I think it's cool. And maybe you guys will have some interest. Looks like all right, looks like we're in action. Sometimes you just have a question or something to add. That's a different rabbit hole. It's okay. You know how much I love jumping down the different rabbit holes. Special. The basement labs have the way better sink and a wider variety of coffee grinds, amongst other perks. Thank you for that enlightening sound, Nippon. Okay, questioning stuff is good. Never just trust random strangers on the internet. I know. I'm a random stranger on the internet, right? Mega Smidge, hello. Oh my gosh, welcome in. How are you? Okay. It's okay, Nippon. You can tease me. I know the mods that is never here is here. I wasn't gonna say it. I was gonna let you call yourself out. I remember, let's see, did I lose a bet with you? And I made you a mod because I lost a bet. I lost a marbles race. Wait, Logic, you know Mega Smidge? Holy crap, what a small world. Oh my gosh, Nippon, you know Mega Smidge? Wait, you guys all know each other? That's insane. Unless you guys are pretending to be. Otherwise, right now, I feel like that person who's like awkwardly like on the outskirts of the party and is like, oh, you guys actually all know each other. Nice. I'm just gonna go over here this way. All right, just kidding. Oh, we all met here? Oh. That's fun. Okay. So I've highlighted the important bits. Should I read the abstract? Is that boring? Yeah, let's quickly read the abstract. So honestly, this is what I do most of the time when I read a paper. I don't want to read like the whole paper sometimes unless I know it's worth it. So a good place to start is the abstract. It's supposed to be easily understood and it's supposed to summarize everything important in the paper. So here, let me get my little highlighter. I think that'll be a little bit more helpful. Should I read this in a British accent? I won't. You prefer vanilla abstracts myself? Really? I like pistachio on mine. Smidge, remember that time with the thing and I went to park my car? Logic. No. I know you were working with the government. Hanford is where we created Fat Man and Little Boy during World War II. Oh, what? I did not know this. The nuclear plant is out there, and I was working for the government and doing patrols. Legitimately heard the rumors of what was out there, just never knew how serious it was. Years later, in a career change, getting the diagnosis and everything really wasn't worth fighting the government for compensation, to be honest. I understand where you're coming from, but I hate that it's like that. Damn, I'm sorry. I hope that I know you mentioned you were in pain, but I hope it's not too awful all the time. Okay. Yes, I would like pistachio. All right, I swear I'm not going to be distracted anymore. Okay, so just a little summary. So a complete understanding of how exposure to environmental substances promotes cancer formation is lacking. This is true. I mean, as I mentioned before on this stream, a lot of the data trying to link in different environmental, like, I don't know, it could be carcinogenic things. It could be like just other substances. In this case, we're going to be talking about air pollutants that are... Are you trying to distract me? Anyways, it's often hard to link these things to actual cancer forming because the amount of time and the amount of exposure is just so long, like years and years before like actual cancer could appear. More than 70 years ago, tumorigenesis, this is the process of a cell becoming cancerous, was proposed to occur in a two-step process. An initiating step where you have the initial induction of a mutation in a healthy cell. So this could be caused by maybe, again, chronic exposure or some type of exposure to a carcinogen, followed by a promoter step that triggers the cancer development. So this could be something that makes that cell that has the mutation now like grow more and more. Or it could make it somehow escape the normal confines of regular cellular growth. So here we propose that the environmental particulate matter, so measuring less than 2.5 microns, this is like common air pollution, known to be associated with lung cancer risk, promotes lung cancer by acting on cells that harbor or have pre-existing oncogenic or cancer-causing mutations in healthy lung tissue. Focusing on EGFR-driven lung cancer, which is the more common form of lung cancer in never smokers or light smokers, we found a significant association between the PM 2.5 levels or the pollution levels and their incidence of lung cancer. For 32,000 957 EGFR driven lung cancer cases in 4 within country cohorts. Okay, this is a lot of people. So they're looking at a pretty large, they have a big N. We like big N's. That just means there's a big sample size. They also did functional mouse models. So this is great because obviously we can't experiment with people because that would be messed up. But there are mouse models that are where we can like have EGFR driven tumors form and we can like do experiments with them. And they do that in this paper. And they found that air pollutants cause an influx of macrophages into the lung, which releases the interleukin beta, which is this inflammatory cytokine. Normally we think of like macrophages and being more important for like, you know, like they need to release these cytokines and things to help kill off like bacteria and other things. And we usually think about them in a good way. But here they actually find that this immune cell that's normally good in our body is actually promoting cancer in this context when there is a high level of air pollutants present. This process results in this progenitor-like cell state. So progenitor, it's kind of like, you could think of it as like a stem cell almost. I wonder if, I think they define this a little bit more later. But you could kind of think of it as like a cell that can just keep growing. It's a cell growth related thing. I'm not explaining that very well. But I actually forgot how they define the progenitor like cell state, so I need to reread that part. Anyways, so they get this progenitor like cell state within their EGFR mutant lung alveolar type 2 epithelial cells. That's a huge mouthful. These are just like cells that line the alveoli. So those are those little pockets inside of your lungs, those little balls. These are the cells that line those, that kind of make up your lung. So these are like lung cells, and they have a mutation in them that is this EGFR mutation, which again, as I mentioned, it makes cells more likely to continue growing because their EGFR receptor is always on and it's always telling the cell to like grow. They did mutational profiling of normal lung tissue from 295 individuals across 3 cohorts of people. And they saw that there was oncogenic, so cancer-causing EGFR and KRAS driver mutations in 18 and 53% of the healthy tissue samples, respectively. So 18% of them had the EGFR mutation and 53% of them had the KRAS mutation. These findings collectively support a tumor-promoting role for PM2.5 air pollutants and provide impetus, which is a funny word, for public health policy initiatives to address air pollution to reduce disease burden, which again, I think as Country Logic mentioned, how are we going to incentivize reducing air pollution, right? There's not really a great financial incentive there. Looks like a pretty normal sized N to me. Honestly, like at least in animal studies, the minimum N that you need to have for statistical relevance is an N of 3. Those are for animal studies, which is a really small number. And it's often argued about a lot by scientists as to like how many samples you need to have. And if I'm still ignoring your mint chocolate chip comment because it is disgusting. I'm sorry. I hate mint chocolate chip. It's one of the worst ice cream flavors. 6, to explain it better, the tumor is between T11 and T12 and basically in a really bad spot to operate since you're walking. They mentioned that the likelihood of paralysis was higher than 70%. And if it becomes an issue, then it will go down that road. But right now it's not life-threatening, just life-altering. Oh, so the pain is basically the vertebrae being squeezed against each other by the tumor, creating a leverage effect. The concern is that in 5 years or so, if the tumor continues to grow, that the cord could pinch and sever. Oh, God. 6. I am so sorry. That is awful. It's like you're screwed either way. If you leave it, you could have your spinal cord get severed, or if you operate, it could also get severed. You're not reducing anything? Wait, what? N of 1 is totally valid. That's how parenting works. Oh yes. You hate it too. I'm guessing that's the mint chocolate chip. I randomly remembered reading about structures that supposedly suck CO2 out of the environment. I know that they have these air filters or some type of filter for plants that are producing a lot of air pollutants, like carbon capture filters, I believe. And that could be helpful. But I mean, also they're arguing, oh, like cars emit a lot of carbon emissions too. How are you going to, you could just put some sort of filter on the car, but like, is that expensive? And then like you have to do that on all the cars and there's already lots of cars on the road. Like what? Yeah. An NF3 is relevant. Well, usually for a small study, sure. Usually you want to do a lot more than that because people won't take you super seriously. And it also depends on your animal model. If you're working with primates, it's obviously very expensive to house and do experiments with primates. So a smaller N is more acceptable for that paper to be published. But if it's like a mouse study, usually you want to have, like per group, you want to have at least 5 to 7 mice per group. And then you want to repeat that experiment, that whole experiment, at least maybe 2 to 3 times to show that the trend is still occurring or to show that the results are like still the same. So, it depends. And then like if you have an even, I don't know. Yeah. Anyways, that's all I'm going to get into. They always use the worst possible chocolate they can get their grubby hands on for mint chip, right? And actually the part that I hate the most though is the mint flavor, that fake green mint flavor. Ugh. Talking smack about mint A, that's a lot of lip coming from someone who's missed most of the 80s and 90s movies. Hey now, what does that have to do with anything? Oh, 6. No, I'm getting my PhD. I don't have it yet.
Welcome to whatever this is.
Yeah, whatever this is. No, I'm in the process. I'm a 3rd-year grad student. And so I'm still a baby. I'm still a baby scientist. So you can take it with a grain of salt, I guess. We've used so many mice in so many labs. We had basically a, hey, I need to have a 20 mice ready to go by June. Can you make that happen, office? An office? Oh, wow. That's wild. No, in our lab, we have like this Excel sheet where we sign up for mice. And there was a time where I was the guilty party that was using a lot of the mice and people were getting upset about it. They were like, we need to breed more mice because CAF keeps using all of them. And there was a time, like literally a few months ago, I kind of hit this point of burnout because I had a lot of stuff going on. I had over 100 mice at one time in different studies that I was working on, and I was teaching, and I was taking a class, and trying to keep account of all these mice. I almost lost my mind. I almost exploded. But yeah, we're past that now. I'm like, I ended all those experiments and I'm slowly starting it again. But I don't know if I'm probably going to have to go to 100 mice or more at some point just to get all the results I need as soon as possible. But it's not fun. And the worst part is that I start to get like carpal tunnel in my scruffing hand. So scruffing is what you call, it's how you pick up mice, because you're like grabbing the nape of their neck. They have like a little fat part behind their neck, and we just like grab that and like pick them up. And this motion, this like pinch and twist, it can really mess your wrist up. And like a lot of senior grad students and even postdocs, I saw that they constantly had to wear these braces to keep their wrist from like bending awkwardly to prevent the pain and like the pinching of the nerve there. It hasn't happened to me in a while, and I'm trying to prevent that from happening. I've thought about trying to do some wrist exercises and stuff as a preventative measure, but it's a side effect of the job, you know, as they say. Carbon capture. Oh, hello, Zelhart. Carbon capture, car exhaust, distill it and spray it into the carburetor. Wait, these are words. I do not understand. What's a carburetor? That's a part of the car, right? I'm guessing. Two arms, grabs pitchfork. Wait, what? What is the pitchfork for? Is this because of the mint? This is why you have interns, Calf. You make them do the paperwork. Oh, it's the paperwork's not the problem. 100 mice. Is your name Calf or Cat? Yeah, I know. I use the dang mice. That's what they're for. Gosh, Mega, come on. No, it's just like a competition because multiple people need them. So, you know, they're a limited supply too. The mice in the little bags are cute. Oh my gosh, Lynx, do you also work with mice? Okay, there's these little like grocery bags. Well, actually, we use takeout containers for our mice. It's so messed up because people are like, oh, is that food in there? I'm like, I mean, I guess, it's depending on your definition of food. The senior project, make a device, do that for you. Oh, the scruffing. You know, I've heard this mentioned before, like, is there a way to automate picking up mice? And it's not that straightforward, unfortunately. But it would be nice if that was a thing. But then again, it would probably be like so expensive that most labs couldn't even like afford it, for example. Gotta watch out for those mice. A friend of mine gave them T1D for 30 years until they got revenge and gave it back. Wait, T1D? I don't know what that means, Colonel. Oh, that must be a poison. Wait, they gave it back? Did they poison your friend? Okay. All right. I've gone down the road of distraction. I think we can rein it back in. Okay. So let's see. Oh, here's a fun statistic I highlighted. I didn't know this because I don't live in the UK. I actually don't know what this is globally, but apparently lung cancer and never smokers, which honestly, this term never smokers makes me think of Neverland, which makes it sound magical. And I'm like, never smokers, but that's not good. Lung cancer in never smokers is the 8th most common cause of cancer death in the UK and has a distinct and clinical and molecular characteristic compared with lung cancer in smokers. So apparently these never smokers, they have this EGFR mutation and it's pretty common. And they're like, huh, that's interesting and weird. Let's take that into consideration. And then here they requote the stats. Air pollution accounts for 7 million deaths per year, with 99% of people living in areas that exceed the WHO's guidelines, which is anything greater than 5 micrograms per millimeter cubed is apparently too much air pollution. And then, oh, here we go. Particulate matter is a key constituent of air pollution and is classified by aerodynamic size. So let me turn the music down a bit because it's getting a little loud. So fine particles that are less than 2.5 microns are able to travel deep into the lungs and are linked to multiple adverse health effects, including heart disease and lung cancer. Because I think there was at some point I was reading about like bigger particle sizes, and apparently those are not able to travel as deeply into the lungs, which is why they're focused on the really tiny little things. Does the UK still heat many homes directly with coal? I know it was a big problem last century. That's a good question. I don't actually know the answer to that. I feel like intuitively, I feel like coal would have bigger particle size than 2.5 microns, but maybe that's not true. Actually, well, no, this isn't really related to coal. This is more related to people who mine other things, but there's this condition called silicosis where your lungs actually get filled with like this fine silica dust, and it makes your lungs look crazy. Like they basically get really hardened and full of and black. I mean, coal is also really bad for your lungs, obviously. Like that's equally bad. We could look at coal lung and silicosis lungs later, maybe. So let's see. What did I want to say here? Okay, let's skip down to here. So they had previously been studying lung adenocarcinoma tumors in a different cohort, and they revealed that even despite a history of smoking, a small minority of patients, so these are people who did smoke, but they didn't have evidence of like a certain mutation that's associated with smoking. And they were like, interesting. And then they were like, consistent with that analysis, in their current study, 7 to 12% of the smokers did not have a driver mutation or a single nucleotide variant that could be attributed to a smoking related mutation signature. I'm not going to read through all of this. Basically, they were like, okay, there's some people, because they were previously studying just smokers, and they noticed that some smokers didn't have that typical mutation that is caused by the carcinogenic smoking substance. Right. The coal lung is called black lung. Oh, thank you for that, Mega Smidge. Dang. You're on it. What about ash? Ooh. Ash? I would imagine you have a variety of particle sizes there, but I know ash can definitely mess up your lungs as well, because if you've ever been breathing in air during a big wildfire, I'm sure you felt at some point your lungs getting really irritated, and that is yeah, that is a big issue. I feel like I read a paper where they were talking about ash. Yeah, but it's going to mostly be bigger particle sizes than what they're looking at here. Okay, so again, to understand whether the air pollutants can promote the formation of lung tumors without inducing any kind of other mutations, they were looking specifically at this EGFR-driven lung cancer, which is really common in England. The probability that a lung cancer in a non-smoker or never smoker is caused by this mutation is like 36 to 40%. Right, 6. Oh my gosh, do we have another ad break? Sorry, it's just now telling me that there's an ad break in progress. Okay, I'm sorry, guys. Take a hot minute here to un-ad break yourself. We'll come back. Let me see. Yeah, it is running. I will just continue going through this though while we're at it. Okay. So let's see. Oh yeah, I wanted to skip that part. So they were looking at a specific time frame of like 3 years of cumulative particle 2.5 exposure, so pollution exposure. And they wanted to see if it was associated with lung cancer in an independent cohort, not restricted to EGFR driven cases. So now they're just looking at everybody, including like people who have the mutation or who don't have the mutation. And this is where they have that big data set of like almost 500,000 participants, which is crazy. Oh yeah. To avoid the ads, you should sub to CAF. Thanks, Ip. You're like my little like the bot command that I need to have, right? How long do I have left? I have probably 3 more years left. Oh my god, Logic. Ash is great for garden potassium. Yeah. If you get silicosis from mining or living near a volcano, you can call it, oh god, links, pneumono ultra microscopic silicovolcanoiosis, if you want. I think people like would condense that into a nice little acronym of like PNMV or PNMSV or something.
Why, hello, my little coffee bean. Welcome to the Caffeine Club.
Logic, thank you so much for the resub. I appreciate it. Aw, I'm sorry I tormented you with the ads now. Angry Moose on the loose. Welcome in. Dork vibes. I'm stealing it. I don't know if you can like physically steal dork vibes though. You have to like remove them, like cut them out of somebody. That sounds really messed up, by the way. Sub to CAF so we can get a coffee giveaway. Lovely Lou, hello, welcome in. Yeah, that's true. Every 20 subs, I do a coffee giveaway, and they're pretty awesome. As I started off my stream earlier today trying some of the coffee I'm giving away, and it's pretty good. Okay, that's a really bad way of selling an idea. Calf won't let me sub? That's not true. You are subbed, aren't you? You are. I haven't heard the word dork in years. Dork is so like 2005. It's not actually a medical term. It was made up for like some kind of Scrabble organization. They just call it silicosis in medicine. Are you serious? That's a wild word. I could see that being a very great word to have in Scrabble because of all the V's and stuff. Every fiber in my body screams to me that slightly in your name is actually hardcore. Oh, okay. Anyways, no, Styles, you are not allowed to drop another. No, absolutely not. Please don't. Please, I refuse. Okay, so let's go back to this. Hang on, where was I at? Okay, so right. So the people that they're looking at in this study are people who have lived at the same address for 3 years, and their analysis showed that the relationship between their lung cancer incidence from this cohort and their pollution exposure levels remains significant. So this is in figure 1D where they're doing a little correlation curve analysis. Where is figure 1D? Hang on, that's figure 2. Hang on, we can find it. All right, I can read the alphabet. This is actually, it doesn't look super significant. If you look at the little blue dot out here, this is like super significant, and these are all from smokers. So if you're a blue dot out here, you have a really high chance of getting lung cancer. Now, if we look at the pollution metric, which is right here, you can see, and also age, you can see that there's like ever so slightly a little bump on the 0 line, but according to their statistics, this is still considered significant. So age, pollution, pack years. I have no idea what pack years means. And then smoking status. These are all risk factors for lung cancer. What the heck is a pack year? Are we like animals now? Like donkeys? Like a pack animal? They don't really talk about that at all, so that's weird. Anyways, so in their cohort, they see that age, smoking, and exposure to pollution is a risk factor for this lung cancer. Cool. Not cool, but cool. Interesting to see that. Now that they have this like sort of now they're going to move into like animal models. So this is where we're now looking only at mice, okay? Dork's current meaning has been corrupted. Wait a second, does dork mean something bad? Have I been using this inappropriately? Oh God, Nippon, tell me, what have I done? Have I committed a crime? I think pack year is 1 pack a day for 1 year. Links. Wait, really? But they have heavy smoking. And then they have like moderate smoker. Dork is a whale member? Interesting. These are all things I'm not aware of. So they're using an animal model where they have these special mice. They have a very long name, but essentially these mice, they have a mutation in their EGFR gene, which again, this is a growth factor receptor. And if you have this certain mutation, then it will constantly tell the cell to keep like turning on its sort of like growth genes and stuff related to that. So they took these mice. So these mice are going to get cancer anyways, but they had a group of mice where they either gave them, they exposed them to particulate matter, which we can go to the methods and see how they did that, or they didn't expose them to particulate matter. And they saw that at 10 weeks after exposure to the particulate matter, they saw a dose-dependent increase in the number of pre-invasive neoplasias. So these are like sort of like pre-cancer. So they're becoming cancerous, and eventually I believe they find them to be actual tumors. And these are in their little figure over here. So let's look at this red circled box. On the y-axis, we have lesions per millimeter of tissue squared. You can see in the control group, we have a very small number of lesions. This is, I believe, by 10 weeks. And then they have a 5 microgram dose of particulate matter and a 50 microgram dose of particulate matter. And now you can see that there's way more lesions in the high dose of particulate matter, as well as even the 5 microgram dose. Although, yeah, it's like barely significant right here. I would say it's a moderate increase. I want to see how they actually give this particulate matter to the mice. Because that's somewhat relevant. Do they even put methods? Do they not put methods here? They do put methods. Okay. Let's see where they do mouse stuff. Wow, these are a lot of methods. Jesus. Animal procedures. Okay. Okay, here we go. For exposure to, I'll highlight it, for exposure to the fine particulate matter, or PBS, which is just actually a liquid, it's just a saline liquid, they resuspended, let's see, the particulate matter was resuspended in sterile PBS using sonication, and the particle size distribution was confirmed using dynamic light scattering. And it had a particle diameter of 2.8 microns. So a little bigger than 2.5. They anesthetized the mice, so they put them to sleep, and then they give them an intratracheal administration of 50 micrograms or 5 micrograms of particulate matter in a volume of 50 microliters. So what they do, this is kind of messed up to the mouse, and I'm going to demonstrate with this pen, okay? So you suck, so they're actually like suspending the particulate matter in liquid, not in air, and then they like take it and they like shove it down the mouse's trachea. It's like a curved needle that they have. Obviously I'm not going to stick this pen in my mouth, but they stick it all the way down to make sure it's like down here in the trachea, and not like in the esophagus or something. And then they like just pump the liquid out, which honestly, I'm kind of surprised that the mice don't somewhat drown from this. It must be I guess it's 50 microliters, so it's a small enough amount. I feel kind of bad for these mice. They did this administration with the particulate matter or the control, just regular saline, 3 times a week for 3 weeks. Wow. With at least 48 hours in between each administration. So that is how they give the particulate matter to the mice, by drowning them. No, I'm being a little dramatic there. Okay. They also saw that there was, let's see, the neoplasia, so the like precancerous lesions, was restricted to a certain type of cell, which are these alveolar type 2 cells. Okay, that's interesting. Let's see. Then they were like, let's find out by which mechanism this particulate matter might be promoting this EGFR-driven lung tumorigenesis. So again, they confirmed that the cells where the cancer occurred in were cells that had this EGFR mutation. They did a analysis of the types of tumor clones. So a tumor is a compilation of cells, right? And each cell is going to be slightly unique, but you might have some cells that are clones of each other. And if a clone is really a good cancer cell, there's going to be more of that specific clone than like other less great cancer clones. So they're looking at like, you know, how many different clones are there in the tumor? What kinds of mutations do they have? And they look at this throughout early tumorigenesis in their mice. I think ET was the EGFR mice. They're just calling them ET mice. And they saw, so let's see. They saw that the expansion of the EGFR mutant cells did not occur during the particulate matter exposure, but manifested in the period after the end of the particulate matter treatment. So they only did this for 3 weeks, and then they waited a total of 10 weeks. So in between the 3-week window and the 10-week time point, so like a total of 7 weeks, that's when tumorigenesis actually happened. So it took a little bit of time. Also, hammy gua 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 gua. Welcome and thank you for that follow. I appreciate it. We're just reading a little bit about how air pollutants can trigger cancer, lung cancer specifically, in people who have never smoked before. And we're reading this very exciting Nature paper that was published last year. It's actually really cool because we're seeing a direct mechanism between like the addition of the particulate matter or pollutant that they're using and the direct triggering of cancer in this mouse model, which is really cool. And it mimics what was seen in a human cohort of almost 500,000 people. Whoa. Okay, dork. Oh my God. Dork means I see. Okay, thanks guys. I get it now. I get it. A mole? Are we talking? Wait, if I'm remembering right, it's like a reference number, like a dozen or Avogadro's number. Oh, I think that now I'm way past that. I don't remember what that's in reference to. So like to summarize, how many total cigarettes someone smokes over a period. So if you did like a pack a week for 7 years, it would be 1 pack per year, but 1 pack a day for 1 year is also a pack per year. Okay. Interesting. Wondering same, how don't they aspirate? Oh, hello, Owl. Oh, you're wondering like how do they actually like aspirate the like the liquid into the lung of the mouse? You know what? Actually, I take it back. I have actually I didn't do it personally, but I saw someone else. I think they were trying to put some virus or something into the lung of these mice, and the virus was suspended in a liquid, but it was a very small amount of liquid, and they did it intratracheally. It's very difficult, and you have to do it carefully so that you don't actually you know, drown the mouse. But yes, okay, I'm remembering this now. It's not a fun procedure, and you have to be pretty good at it. I am not. I haven't personally done it though. Reminds you of the liquid oxygen scene of the mouse in the abyss. The mouse in the abyss? Is this an anime? Sorry, Nippon. Man, them scientists are cool psychos, but someone's got to do it. I mean, it's done while the mice are asleep, Mega Smidge. They don't feel a thing. Someone grew an ear on a mouse. I think it was ear cartilage. What? Wait, what? That's wild. That seems like a lot. I wonder how the method compares to aerosol particulates and if that would change the results. Oh, how does this method compare to like aerosolized particulates versus the liquidized particulates, and would it change the results? I would assume that it wouldn't change the results because in either case the particulate is still getting into the lung. But yeah, I mean, there are ways where you can give the particulate through an aerosolized method. I've seen that in other papers, but I think it's not as easy to do, and you have to like actually fill like a little chamber up and then put the mouse in it and then pump the particulate into the chamber. And I think it's a little bit harder to control how much of that particulate is actually being taken up by the mouse versus here, you know that exactly 5 micrograms is going into the lung and exactly 50 micrograms is going into the lung because you have that controlled amount in the liquid. So it's more quantitative and controlled to do it the liquid way versus the aerosolized way. But you could definitely put the mouse in a chamber and just pump the particulate in through there too, which is how a lot of older studies have done this with other things. Yeah. Okay. So let's see, what did they find? They saw that the particulate matter seems to work in 2 ways to promote this early tumorigenesis. One, by increasing the number of EGFR mutant cells with the potential to form a tumor. So they haven't yet, they're just a neoplasia. And by increasing the proliferation rate of these mutant cells within the early lesions. So they're proliferating faster. So then they wanted to test whether the particulate matter induces DNA mutagenesis. So does the particulate matter somehow directly like get taken up by the cell or cause the cell to have more mutations in its genome? So they can do this by doing whole genome sequencing. So they're literally taking the cells, they're taking the DNA out of the cells, which represent this entire genome of the cell, and then they're sequencing it. And this is like it's somewhat expensive. It's a pretty expensive process. And then they look at literally every segment of the DNA to see if there's more or less mutations in the DNA taken from the cells that was in the mice that got the particulate matter. And they found there was no increase in the number of mutations in the tumors from the particulate matter exposed mice. And this is in figure 3. I think this is going to 3C. Let's see. Wait, was this figure 3C? 3A through C. All right. Oh, I hate these PCNA plots or whatever. I do not like these. These are okay, what are they trying to show here? Wait, is this actually figure 3? It is. Okay, this is a PC analysis plot of RNA-seq data. That's not it. Wait, what? This isn't the sequencing data. I'm confused. Heat map. Wait a second. This is not the data they're saying. Oh, extended data. My bad. This is in their extended data. So that's somewhere else. I didn't actually download that part of the PDF. Anyway, so we're just going to have to take their word for it. They didn't see an increase in mutations in the mice that were given the particulate matter. So that's not what's causing the cancer or the increased rate of neoplasias in the lungs. So then they were like, well, what if the immune system is doing something to cause the cancer? So what they do is they cross their special EGFR mutant mice to mice that have no B, T, or NK cells. So these are like mice that have no immune system, mostly. They still have their macrophages though, and they still have like neutrophils and other things. And actually, this is where it gets interesting because they pin the macrophage as the immune cell that is responsible for causing this cancer. But in this immune deficient model, where there are still macrophages, they don't see an increase in neoplasias. And this is kind of confusing to me because I'm like, the macrophages are still present. So how are they like not causing the neoplasia when you have your TB and NK cells gone? And there's probably some important little detail that I was just missing, but it was a little bit weird to me. Anyways. Oh, it's okay. The Abyss is a 90s movie. Oh, I see. Oops. My bad. I was born in the... No, I should know these old movies. I just don't. Spleen! Hello. You know, I always call you Spleen, and I actually don't know if your name is like P or Speen. I don't know. Anyways, moving along. You maybe a control group that got the liquid without the particulate. Oh, having a control group like that? Yes. Well, they do have that. They have the PBS groups. So they had a PBS. So PBS is a liquid. It's like a saline liquid. Or no, is it saline? It's basically like a neutral solution that we use for everything because it doesn't really have an effect on the mice. And they either resuspended their particulate matter in the PBS or they just gave the PBS by itself to the mice. And that was their control group, was the PBS by itself with no particulate matter. So in that case, it worked as a good control. Angry Illinoisan. Wow, I'm sorry. I said your name so wrong, but welcome to stream. I hope you're doing awesome and having a good Saturday. Welcome in. We are just talking about pollution, particulate matter that's aerosolized, and when you breathe it into your lung, it can increase the risk of cancer and how that happens. The mouse was breathing frantically while liquid oxygen flooded its lungs. Its breathing became steady once it got used to it. Oh, this is, oh, I see what you're talking. Oh God, that's awful. Oh, this was a scene where the diver needed liquid oxygen to dive deeper. To show it was safe, the one proposing it took one of the crew's pet mouse and closed it in the liquid oxygen. I see. Okay. Very interesting. Okay, so let's see, is the immune system necessary? So in contrast to the mice with the intact immune system, they saw that exposure to the particulate matter after they had their mutation happen. Hang on. What? I'm good. So they saw that in the mice that don't have an immune system, these immune deficient mice, they did not see an increased rate of cancer. This suggests that a competent immune system is required for the particulate matter enhanced EGFR mutation driven lung tumorigenesis. So basically one of these components of the immune system is needed, something is being triggered that causes the cell that already has a mutation to actually become a fully fledged cancerous cell. Wow, I pronounced that really weird. So then they have a little hypothesis here. They're like, okay, well, we know that inhalation of toxic particles can induce a local response in the lung, which is mediated by macrophages and lung epithelial cells. Again, I'm a little bit confused here because they didn't specifically like knock out macrophages or like remove the macrophages. So I don't know how they can say that the macrophages just weren't present in their immune deficient model because it wasn't totally immune deficient. I think maybe the macrophages in the immune deficient model, they might have a dampened response because they're missing a key cytokine gene, which is knocked out in that model called IL-2, but it's not really required by macrophages, so I'm a little bit confused by that. Anyways, they're like, okay, well, let's look at macrophages now. And so what they do is they see that they look at mice or lungs from control mice. Let's see, wait a second. They see more dendritic cells. Okay, here we go. They see there's a greater density of macrophages after the particulate exposure, both acutely, so right after they pump it into those poor little mousy lungs, and 7 weeks after the exposure. So these macrophages, they come quickly and they hang around for up to 7 weeks, which is not great. They identified what these macrophages were based on their CD protein markers. We're not going to get into that. They also observed an increased number of macrophages in other models. So they had another EGFR-driven mutant mouse model, and they also had the KRAS mutant mice. So again, KRAS and EGFR are the two main mutations that pop up in humans with lung cancer that is not associated with smoking. And that's why they're using these mouse models here. Okay. All right. So now, let's see, they're looking at the lung epithelial cells. Do they go back to the macrophages? Okay. Oh, right. So they were looking at some of the cells because now they're like, okay, well, why do the macrophages come into the lung? Maybe the lung cells are releasing something that recruits the macrophages to the site of the particulate inside the lung. And so they find that in the mice, particulate exposure led to an upregulation of genes in the epithelial cells that are known to regulate macrophage recruitment, including those that encode the IL-1 beta, which is that inflammatory cytokine. And then there's some other proteins that we're not going to talk about. But basically, these things kind of help bring in the macrophages and make them function better. Okay. And then, oh, actually, did they have a figure for that? Let's see, figure 3B. Let's quickly look at that real fast right over here. Oh yeah, this is, okay, this is the thing. So what they're looking at in this plot is gene expression. So genes encode for proteins. And if there's a hot red block, that means that the gene is turned on really high. And if it's like dark purple, that means the gene is sort of depressed and it's not turned on. So they're looking at genes that are turned on in the condition where they have, I think these are normal mice, and then these are mice that have particulate matter and the EGFR mutation. And you can see that a lot more of these genes are turned on after mice are treated with the particulate matter because it's all red. So the genes are turned on pretty high. And then in their control group, you can see that the genes are all sort of bluish purple. So they're like turned off or they're not being expressed that much at all. That's my sloppy drawing. All right. Moving along. Okay. Right. Okay, so these results identify these particulate matter induced transcriptional changes or gene changes that we just saw in the lung epithelial cells that are associated with inflammation and lung progenitor cell states. So they wanted to test whether these alterations in gene expression are also reflected in functional differences in their epithelial cells' behavior. So they isolated the lung epithelial cells from these mice following their exposure to the particulate matter. So now they're just like taking them out and they're culturing them in a little dish in the lab. And they do this 3D lung organoid formation assay with their little lung fibroblasts. And what do they find? They found that in wild-type normal mice exposed to particulate matter, there was no increase in the organoid formation. So this assay, what it's testing for is if a cell is truly cancerous, it will start, it doesn't have the normal inhibitions of a normal cell where typically cells, they grow in a monolayer, so they grow flat. But if a cell becomes cancerous, it ignores these rules and they'll grow in sort of like a little pile. And this assay is testing whether they form a little pile, and it's called a lung organoid formation assay. So normal mouse, like their control group, did not have any formation of these little tumors. However, in their EGFR mutant cells, they saw an increase in the amount of little organoid things that were formed, and that's in figure 3D. I think they might have a cool picture. It's actually kind of a weird picture. So the little organoids are these like little red bumps here. So there's more of them apparently. Although I have to say, like looking at these pictures, it's not impressive until we look at the quantification of it. So if we look at the control group, we see like, okay, there's a very small number of these little piles. However, when we add 50 micrograms of the particulate matter, which is quite a lot, we now see that there is a modest, significant-ish increase of the formation of these little piles. So these cells have been transformed. They're like cancerous now. Ooh, we have a meat stick. I'm going to save that for a moment. Okay. So then they were like, okay, what is... Let's see, did we finish that part? Yes. So now they're asking, what does the particulate matter directly do to the macrophages? Their hypothesis was that the lung macrophages, they release inflammatory cytokines when they're exposed to the particulate matter, and this might be important for tumor formation or promotion. So they take these like little, they take those epithelial cells that they identified as being important for producing IL-1 beta, and they take them out of the mice that are not exposed to particulate matter, and then they induce the expression of the mutation outside of the mouse. So now they're doing everything outside of the mouse, and then they culture these cells with macrophages that have been exposed to the particulate matter inside the mouse or not. So they're taking just like normal, like they have the normal epithelial cells out here, and then they're like collecting macrophages from mice that either were treated with particulate matter or just PBS, and they're seeing like, okay, their assumption is if the macrophage like took up the particulate matter and it's that particulate has done something to the macrophage, if we take it out of the system and put it just in a dish where there's no other variables, will we see it cause cancer in our little like epithelial cells here? And here you're like, this is sort of a reductionist approach where you're like taking out any other variable from the in vivo system, isolating it, and just looking at the things that you care about, which is like your macrophage and your epithelial cell in a dish together. And this is really a cool experiment because they see that the particulate matter exposed macrophage increased the OFE, which is just like how tumorigenic the cell is. Yeah, it increased the amount of like little tumor piles in their EGFR mutant cells compared to the control, and that is going to be in figure 3F. And I think this is probably one of their most important experiments. So if we look right, oh wow, let's not look at this image because it's really it's really sad. Like there isn't a huge difference. But if we look at like the quantification of it, so again, blue dots is control, red dots is like the particulate matter that's been added to the macrophages. We can see that there is a pretty significant increase in the amount of like little colony, like the little tumor piles, when you throw macrophages that have particles onto those cells. So clearly the particulate matter is causing the macrophages to emit something that is cancerous, or that's causing the cell to become cancerous. And then they go on to find out that this is I think it's like IL-1 beta, and they do some other things, which we're not going to go through. But that is basically this entire paper, which, yeah, it's only 4 figures. It's pretty short. I think they go on to like try to apply this back into patients and stuff, and they find more cool things. But I will put this PDF with my little highlights and my little notes, which are kind of messy, into Discord, so you guys can like see it if you want. But yeah, that is it. Which now I'm gonna take a quick water break because my voice is getting parched. Let's see. That's the main. That was pretty much everything. Let's go here. Yeah. Anyways, I thought their experiments were really nice, and for the most part, and it was a interesting finding because usually we think about immune cells as being helpful and fighting off cancer, but here the immune cells are actually promoting the formation of cancer, which is a bit unexpected and interesting.